Our next talk will feature the importance of how dietary microbes play a significant role on human health. Our presenter, Dr. Bruno Pot, a microbiology scientist who's the science director at Yakult Europe, and has always been fascinated by the impact of microorganisms, sorry, I get the same thing, and the impact of microorganisms and for improvement on human health. Bruno believes that the science supporting the positive impact of live microorganisms on human health has reached a point that warrants, a formal communication to the consumer. So Dr. Pot is co-leading work currently co-leading work at the IPA for Dietary Microbes, which has recently seen other collaborator associations join, IFANS and ISAP. The creation of a formal category of dietary microbes can be an attractive option in underlining benefits to consumers. Bruno, the microphone is yours.
If I find how to move this. So what I'm going to talk about is about dietary microbes as announced, but the focus will be on fermented foods more in general. So I will discuss a little bit about the definition. Then one of the observations is that we are consuming less and less of those live microorganisms, and that has some health consequences. So what can we do about that? And also how can we communicate better about that? So what are dietary microbes? Well, if you ask Google, you won't get a lot of hits. I did this the day before yesterday, 1,115. So no definition at all. So I took the liberty of producing my own definition, which I propose here in front of you. Live microorganisms ingested as part of a normal diet. This would include bacteria, archaea, yeast, fungi, both foods and food supplements, of course, but exclude indigenous or oral microorganisms because this is a substantial part actually of what we consume. We consume like 10 to the 10th of microorganisms in this way. But of course they are not through the food. And also it would exclude dead bacteria. I'm not saying that dead bacteria cannot have a health benefit, but at least they will not actively interact with the microbiota, which is part of the health effect I will discuss later on. So if you look to this definition, it pretty much fits with the live cultures definition that was produced by this ISAP committee in 2014, which covers probiotics and non-probiotics. But of course, in this case, we would exclude probiotic drugs because they're not part of the diet. Also, the non-oral probiotics, as mentioned. Maybe we should exclude feed. This is a point we can discuss later on. And definitely, we will not include the FMT, the fecal microbial transfers. So what we will discuss more closely today, as I mentioned, is the fermented foods issue. So fermented foods have been defined quite recently. So in 2017, actually by a group around Maria Marco, as foods or beverages which are produced through a controlled microbial growth and the conversion of food components through enzymatic action. This definition was recently revived, I would say, or slightly modified, in which the word control was just replaced by desired, and the definition remained the same. That was done again by Maria, but in the frame of an ISAP working group. This is interesting definition if we talk about dietary microbes, because it recognizes and it confirms the essential roles of microorganisms in making fermented foods. But what is different with the dietary microbes is that this definition doesn't require the bacteria to be present nor viable at the time of the consumption. And that's quite a difference with the dietary microbes. So what are these benefits now? So we're discussing health benefits. I know like said before, I'm preaching to the choir here, so I'm not going to details to this list. Actually, the presentation will be made available afterwards, so if you want to have, because later on there will be a lot of references coming up, so if you have any interest in checking them, they will be available. But what I want to stress here is if we talk about health benefits of fermented foods, we actually have to discriminate two levels. So the first level is the benefits during fermentation in the food, and then we're talking about not only taste benefits, but biopreservation effects, bioavailability of some minerals, the production of proteins or bioactive peptides, that kind of things. But the point that I want to discuss more today is really the second level of benefit, and this after the ingestion of the fermented foods, what can the microorganisms do inside? And so this is a graph I stole again from the article from Maria Marco that was published earlier this year and merged it with some other information. The references are there. And what is important here is that, well, it's important, and at the same time, it's some kind of an issue when we want to study it, is that you have benefits at different levels. So the first level, of course, which is very important if we talk about non-communicable diseases, is at the immune level. So the microorganisms that arrive in your intestine will interact with the microbiota, and we know that the microbiota has a lot of interactions with the host, which will help to form, or in a positive or in a negative way, the immune system, which could lead to allergies in the case it's a negative interaction, which could lead to allergy or autoimmunity. There's also, as you can expect, if the microorganisms arrive alive in the intestine, a microbiological effect, which is enriching the diversity and is also contributing to probably some of the metabolism which is happening in the intestine. And these metabolites could also have physiological benefits for the host. So it's a complex system, which is very difficult to grasp because it's in interaction with a very complex ecosystem, which is already there. So if we go back to the history of these health benefits, then this is also a paper that all of you, I think, have seen and read in the past. So this is the famous basis of the hygiene hypothesis, which is an idea that was published by Jean-François Bach in 2002, saying that since the 50s, because of vaccination, because of antibiotics, because of increased hygiene, we have had less and less contacts with pathogens. And as a result, in the same period, we have seen an increase in the development of non-communicable diseases like type 1 diabetes, asthma, and Crohn's disease. Now, the idea behind this is that we need those pathogens actually to mature the immune system and to keep the immune system alert. Now, that theory was actually not more or less was not refused, but was modified in a sense by Francisco Guarner, who is giving a lecture, I think, in one of the other rooms right now. And who, together with another group of experts, actually modified that concept in the sense that it is not the pathogens that we need in order to mature the immune system, but that we have developed over several hundred thousands of years, a microevolution, a microecology as a host with our bacteria, and that it is actually a good functioning of this holobiont system, as it has been later called, that is important in maintaining a good metabolism and good immune functionality. So the fact that we have, over these years, had also reduced exposure to these microorganisms, whether they are from the food or whether they are from the environment, has been one of the reasons why the immune system is maybe less efficient and why we see this increase in immunodysregulatory disorders. And so these microorganisms, for that reason, in the paper have been called the old friends. So they even showed with this graph how this could be explained, and actually it's very simple to understand. If you have a pathogen, if your immune system encounters a pathogen, it will evoke a pro-inflammatory reaction, which has the purpose to eliminate the pathogen. However, if you have a contact with a commensal microbiota from a food, the purpose is not to eliminate it. It will evoke an immune reaction, but that immune reaction will be rather anti-inflammatory in nature. You can see if the mouse is there. And it will evoke a regulatory response. And that response is actually evoked through dendritic cells, immature dendritic cells. When they encounter a commensal, they will mature, and they will mature into what we call regulatory dendritic cells, which have the capacity to transform T cells, naive T cells, into regular T cells, which may have an effect in the case of allergy and also in autoimmunity. That is purely immunology. There's nothing new about that. So this theory from 2006 has been confirmed by many other authors in the meantime. So 2016, 2017, saying that it's not about learning what to attack, but learning about what to tolerate. Or we need exposure to a diverse range of friendly microbes, not infectious pathogens. And I think this message is, of course, still valid until today, and is being supported by more and more science. So what then are the consequences if in our Western world and our Western diet we are consuming less of those fermented foods? It's a bit strange for me that we consume less of those nice foods, but apparently it is the case. So the effects of that maybe can be estimated or can be compared to some historical data, as well as to the fact that we still have within our world areas where fermented foods are consumed in a higher number or a higher amount, or also where the contact with environmental bacteria is much higher. And I like this example of the Hadza. This is a population in Tanzania, about 1,000 people still, which are probably the only full-time hunter-gatherer communities which we still have in Africa. They live quite apart, so they don't have many contacts with the society. But in this study, the authors managed to actually participate 27 of them in a trial in which their stools were compared to 16 Italian urban controls. And what you could expect, of course, is that there is a higher level of microbial richness and biodiversity in their stools, which has been linked to the fact that this community, they consume about 50 grams of fiber every day. This is about 3 to 4 times, 5 times what in our Western diet we would consume on a daily basis. So the diversity is one different thing, but also the composition is different. And what we saw, or what was published at least in this paper, is that there is an enrichment in Prevotella and Treponema. Maybe quite unexpected, at least for me, there was an absence of bifidobacteria, which we consider as the beneficial bacteria in the gut. And there's also an increase of Clostridiales. And I assume this is the good part of the Clostridiales, because a lot of Clostridia have a more pathogen background. But these Clostridiales, these bacteria which had the ability to actually digest or extract valuable nutrition from the fibrous plants that they were eating during their hunter-gathering activities. So altogether, this definitely supports the fact that we have developed a microbiota that is functional in function of what we eat and what we encounter of microbiota outside. So this bacteria-host interaction, the holobiont theory, is definitely something that is supported by this kind of observations. Now, I've mentioned this single one, but there is many of those publications out there. So you have the Malawi, Venezuela population compared to people in the US, diet in the US. You have the Burkina Faso example, the Colombian example, the Indian example, Mediterranean diet, which we all know has some impact on the microbiota. So there's plenty of literature actually that all goes in the same direction. What you eat will determine what your microbiota will look like, and therefore also whether you remain healthy or less healthy. So all these observations led, and this is also a paper I would like to mention particularly, all these considerations led to a new hypothesis, which was called the multi-hit hypothesis, which was published in 2017 by Erika and Justin Sonnenberg. And what they actually meant with the multi-hit hypothesis is explained in the graph from the paper very clearly. And it means that like 10,000 years ago, when there were indeed still more hunter-gatherers than there are today, the diversity of the microbiota was actually very high. When these people started to settle down and started to cultivate crops, they actually had a less diverse contact with the environment and with also the food. So the diversity in that period has reduced of the microbiota. Then many, many, many years later, so we come in the industrial time, the industrial produced food, again, because of the processing, had probably less bacteria, and therefore also, again, we saw that there was a decrease in the diversity of the microbiota. And then finally, in the last 100 years, you had many effects that were hitting the microbiota. That was a change in diet. Think about change from direct to fast food, for instance. Sanitation, so the hygiene conditions became much better. Antibiotics became used, but also C-sections and formula to replace breastfeeding all may have had an impact actually on the diversity. Now, this led the authors to say, well, we probably can reintroduce these microorganisms by increasing in our diet the microbiota accessible carbohydrates, the so-called MACs, together with the microorganisms. So that was their hypothesis. Now, we're very lucky this time because last month, only the result of a clinical trial by the same authors actually was published, and the results are quite fascinating. So what they did is actually they included in their study, clinical study, 18 people, which for a period of 10 days were consuming a high-fiber diet 18 people were consuming, same period, a high fermented food diet. There was 3 days of run-in of baseline measurements before, and 4 days after the study in which people were allowed to eat whatever they want. And they used really sophisticated multiomics technologies to look into the microbiome as well as to the host. And the factor in the host was mainly the immune profiling, which was looked at, which is interesting in the light of the discussion of today. How can we improve the immune system and reduce allergy or autoimmunity? And the results, as you can see, are quite surprising in the sense that what they expected, actually increasing the max, did not result in an increased diversity of the microbiota, although there was a switch in the metabolism towards microorganisms that had the capacity to degrade the increased level of carbohydrates and the production of short-chain fatty acids. Looking in this group with high fiber diet, there was almost no influence on the immune system either. So they discriminated actually 3 groups. So one group had a very low diversity at the baseline, and they actually had an increase in the inflammatory markers, while 2 groups that had an intermediate and a higher diversity at the start, they actually had a lower reduction of inflammatory markers. But the interesting part, so the conclusion there is increasing fiber intake in this short period of time, we should be honest, 10 days is really a short period of time, was insufficient to increase the microbiota diversity. While in the group that consumed the increased fermented food diet, there were surprising results in the sense that the microbiota diversity increased from the start in all of the people included, but also the immune markers were significantly changed, and not 1 or 2 markers, but practically all the markers they looked into. That were 4 types of immune cells measured by 13 different immune cell signaling parameters, and also 19 different inflammatory proteins measured from the blood, including IL-6, which is traditionally linked to inflammation, rheumatoid arthritis, type 2 diabetes, or chronic stress. Now, this was interesting, but of course it was not in agreement with what was published before. It was rather surprising because only 1 year earlier, this meta-analysis and systematic review was published, which said that the link between fermented foods and inflammation was not supported by measurements in IL-6, which was one of the markers that significantly increased, neither in C-reactive protein, but although in that meta-analysis there was a slight impact on lowering the levels of TNF-alpha, which is an inflammatory cytokine. So this represented actually 1,426 people in 26 studies retrieved from, let's say, more than 3,000 documents. So what to do with that? So there is definitely conflicting evidence, although there is probably a very good sensitive rationale behind the use of these fermented foods. And what we need, of course, is more studies in order to support the different views there. So one of the studies that I find interesting that definitely could contribute to a solution or to a better understanding, I should say, of what's going on is this study in which more than 6,000 stool samples have been analyzed from the American Gut Project with a multi-omics approach again, so meaning we look at the genes as well as into the metabolites. But these samples were specifically selected from 115 individuals which had a high fermented food intake specifically. And the monitoring was done for a period of 4 weeks. So the results showed that indeed there was an increase, subtle but statistically significant increase of the diversity again, the beta diversity, and the differential taxa. What was interesting is that it was also linked to some metabolic markers. So in this case, they saw an increase of CLA, conjugated linoleic acid, and they could support it based also on the genetic material, that this could have been driven by the taxa which are associated with the fermented foods, namely the lactic acid bacteria, the bifidobacteria, and the propionibacteria. Of course, this is only one study and needs to be increased in numbers and definitely repeated also in other populations for sure. Another study I wanted to show again from 2020 is also interesting in its approach. So what they wanted to do is show the link between the fermented food and the microbiota in a much clearer way. So in this case, 9,445 shotgun metagenomic samples from human stools were analyzed for their content, and from these metagenomic samples, close to 155,000 bacterial or microorganism genomes were created. They compared this genomic content to the 666 metagenome assembled genomes from microbiomes recovered from fermented foods in order to match these two. And in order to know what they were talking about, they included in the study like 200,000 reference genomes of microorganisms present in reference databases. So the results showed that indeed there is similarity between the genomes isolated from the food as well as the genomes isolated from the microbiota of the volunteers here. And this was really substantial, so it seems that 52.4% of the food max actually could be recovered from the stools. In total, it went about, the number of species was 20 species of lactic acid bacteria and 17 species of non-lactic acid bacteria, which were recovered in this way. Interesting that this is not particularly people that consume high amounts of fermented food. So this was just a random study done on stool samples from a collection of probably non-related even metagenomic studies. So what else can we provide as proof? So this is a list, and I don't go to the details, so the presentation will be made available, anybody interested. But this is a list of a number of more recent studies in the last 3 to 4 years that have tried to understand better the link between allergy, for instance, and the quality of the microbiota. So there's a lot of research going on. You can find a similar number of papers probably in other fields, in autoimmunity, in diabetes, and in many of those particular diseases, non-communicable diseases. So what we have, or what we need, is further research, of course, in this trend. We need to use modern technologies that we have. So in order to understand, to also explain how this could be, how it could be that food microorganisms can directly affect health of the host. We need a regulatory approach as well, because currently today the category of the dietary microbes doesn't exist. I'll come back later in the talk on this. And of course we need to confirm the findings with clinical studies, as has been done and published recently by the two authors that I presented. Am I in time still? Okay. So what do we need? Actually, we need a multidisciplinary approach. So fundamental research, collecting enough data, metagenomic data, which come from different areas, from different geographic locations, from different backgrounds, different ages. Use maybe artificial intelligence actually to make sense out of this large amount of data. Then do the confirmation. If there is any association that is suggested, we can try to actually confirm this in clinical setting. We need to try to understand the molecular mechanisms as well in order to be able to explain also to regulators that this is really worthwhile what we are talking about. And actually, I also think that we can get quite a lot of supportive information from geographic work and from paleogenomics work, which shows that indeed there has been a decline in the complexity, the diversity of the microbiota over the years. So if we want really a food category, dietary microbes, to be created, which is probably in line with dietary fibers, it would have many benefits. The benefit is that dietary fibers is something which is known by the consumer as being health beneficial. It is in nutritional guidelines. Dietitians will advise you dietary fibers to increase your dietary fiber content. So this allows us a communication about benefits of food, avoiding the word probiotic. And this morning, this afternoon, Louise suggested that, leave out the health aspect from the probiotics definition. Actually, if we talk about dietary microbes and it is automatically linked to the health benefits, we don't even need to use the word health in the definition. The definition that I proposed didn't mention the word health at all. So there's definitely a possibility. On the other hand, if we want this to be accepted by the regulators, knowing the skepticism that is out there, we will need to make sure that this is science-based and it really holds ground. So this brings me to the last point. We need to do a good job in communication. We just heard a very nice talk about how important communication and education is. This will definitely also be the case if we want to talk and start talking about dietary microbes. So the communication we want to do is quite simple. the fermented foods can impact our microbiota. Our microbiota, as well as the fermented foods in this way, will also promote health. So also this morning there was a mention about GRC. I have a slide as well as this by independence. But what is important, I think, is that not only the scientific community is communicating about it, but also the authorities, which really have an impact. And I'm very happy to see that EFSA published in 2020. Apparently there was another presentation this morning while we were here that mentioned this paper, which states, so from an official organization, that exploring the need to include microbiomes into EFSA scientific assessment would be really something to consider. That means that EFSA realizes that the microbiome actually does impact on health. And actually in the paper it is mentioned specifically that this is with a view to understand the potential health impact. Because one can think that you want to look into the microbiome from the safety point of view, but in this case it's also explicitly mentioning the health benefit. And this report that was mentioned by Espen this morning is very interesting as well, really worthwhile reading, because they discuss the most recent information available on the vital role of the human gut microbiota and associated opportunities for human health and well-being, and also that this will have an impact on healthcare, nutrition, and well-being. So it goes even much further than only the health, but also there's economic benefits that can be expected from this. FAO already in 2010 published these 4 dimensions related to sustainable diets. If you read this document, the fermented foods clearly fit completely into that definition and supports all the 4 dimensions in this, knowing specifically that by 2050 we will have to feed 9 billion people in this earth. Initiatives like the ones that have been taken by California Dairy in fermented foods and nutritional guidelines, global foodomics, a crowdsourced window into microbes in our foods, then this website that most of you probably have known or have consulted at a certain point, Your Guide to the Difference Between Fermented Foods and Probiotics. That's an interesting one. Because then how do you fit probiotics into the category? Well, the answer is how do we fit prebiotics in dietary fibers? It's just a subcategory. So probiotics could be considered as a subcategory actually of dietary fibers. And then there's also ISAB that has shown in the past to be interested in the topic. So one last thing is now is the time to use the opportunity, use the moment. Because in 2016, fermented foods were already one of the top 10 food trends. And this year, in 2020, according to this source, the fermented foods are currently the number 1 of the 10 big food trends that are ongoing and are exciting and inspiring. So fermented foods on the top. And if you go to social media, you will find proof of that. So the 7 must-eat fermented foods, the 8 fermented foods and drinks to boost your digestion and health. And if you want to eat more fermented foods, you already get for free 2 recipes that will help you. And we can see that indeed people are considering to make their own fermented foods at home, the kefirs and the yogurts for sure. So what are the ongoing initiatives? As I mentioned and as George mentioned, IPA is actively involved in trying to establish a committee of experts. We had 3 meetings in the meantime, in which we considered to create a dossier about dietary fibers, about dietary microbes that matches the dossier for dietary fibers, and which could be proposed to EFSA for a scientific assessment. It doesn't mean a health claim or a nutrition claim. It's more an assessment of a food category and see what the reactions could be. ILSI North America, now IFNS, also has an interest through their Gut Microbiome Committee, and they actually work together in the USA with USDA. They recently got a number of additional questions in the USDA database asking, does your food contain microorganisms? How many? And how are they being counted? ISAB has a similar interest in the US. They have already published this the fermented food paper that I mentioned earlier in my talk. And they continue their interest through the ENHANCE database in the US as well as in Korea. LABIP, Lactic Acid Bacteria Industrial Platform, within 2 weeks in Bruges is organizing a workshop on the technology of fermentation and on the benefits mainly in terms of biopreservation. Also, of course, there is the many different research initiatives worldwide using these fantastic omics technologies which allow us to really compare and understand not only who is there, but also what are they doing there. This is also helping, of course, to better understand what's going on. So the more people definitely are interested to contribute, I think also the more credibility the topic might get if we would go ahead and try to create this category. So the initial question, can we afford not to think about dietary microbes more deeply? I will leave up to you to answer that question, and maybe if there is other questions during the roundtable, I'll be happy and try to answer them. Thank you very much.